everybody, welcome to the next instalment of the Mini Medical School. So tonight we've got 3 speakers talking about COVID-19, containment, treatment and vaccines. We've got Matthias, Schmid, we've got Christopher, Duncan and Ashley Price, all doctors from the NHS. So I'm going to pass over to Matt to start. Just so everybody knows, you can use the Q&A function throughout and we will ask questions at the end to the 3 speakers. Thank you very much. You can get going, Matt.
Thank you, Laura. Good evening, everybody, and welcome to this talk. And I hope that after this talk, you'll all be interested in wanting to study medicine and especially are interested in infectious diseases. So normally I would ask you, what is it about infectious disease and what is COVID is an infectious disease. And so what do? Most people think we look at agar plates and we prescribe antibiotics and we seem to know really strange names like paracoccidioidomycosis, but really we are interested people who look after the most fascinating and new infections. We are at the cutting edge of treatment research. We help prevent diseases. We help prevent spreading diseases. We look after patients, not just from an infection, but holistically. We look at them scientifically. We show compassion with all our patients. And some of us have even great knowledge in their specialty. We also look after coronavirus infections at this moment in time. 2 of them are highly infectious, and actually this one, COVID-19, started off as a highly infectious disease as well. And as you can see, we all like a good drink, and particularly special drinks like this one that I've put up here. So how did this all go on about? COVID will teach us a few things. One is about containment and how good what we need to do. And one is about pandemics. The oldest pandemic that we really properly know about is the Black Death in the 14th century. About between 75 and 200 million people are thought to have died, which equates to about, at that time, around 1/3 of the world population. So just imagine that 1/3 of you would now be dead or would be dying within the next few months. So that is quite an astonishing figure. But big outbreaks we've had over the years were particularly the Spanish flu. That was cost more lives in 1918 than World War I to get all the deaths of World War I. And then we've had huge outbreaks of measles, HIV, smallpox, and now most recently, of course, COVID-19. What this had taught us in the 15th century, we used very fancy methods of how we tried to protect ourselves. So it was the start of what was called PPE or personal protective equipment. This had developed and luckily, so we're pleased that we don't have to wear these inventions from the 15th century anymore or the 16th century, but we're now wearing much better equipment. And there are 2 types of equipment. One is what's called a personal PPE, and the other one is based upon an even more advanced method, which is a power assisted respirator system, and I'll show you that. So we here in Newcastle, we have an infectious diseases unit, and we are a little bit unique because we became one of 2 centres in the country, actually in the 1990s when we were still at the General. And then when we moved to the RVI in Newcastle, we started thinking about rebuilding a unit, and it was quite complex. We were put on standby during the SARS outbreak, and I'll tell you what that was. And we were then in 2014 faced with the problem of a worldwide pandemic or nearly pandemic called Ebola from West Africa. In 2015 also we had an alert for another coronavirus infection that was MERS, and since then we have every year alerts on that. And we are still to this day one of two national units looking after highly infectious diseases like Ebola or viral hemorrhagic fevers. And luckily, I have to say, within the UK setting, this is an extremely rare event. Otherwise, if you look at sometimes the news, what happened in Sierra Leone or most recently in the Democratic Republic of Congo, this is really quite a really bad disease that kills still 60 to 70% of people out there. Just to show you the evolution of what's been happening in 2003, and that is probably for some of you older than you are, we had a new disease emerging and it was not known at all. And it became ultimately a disease starting from the sort of southern China area and then moving very rapidly all over the world, or in most areas of the world. And it was named SARS, which stands for Severe Acute Respiratory Syndrome. And it has had a high mortality rate. Indeed, it cost a lot of healthcare workers' lives. There were theories about this, that this was actually a conspiracy theory, that it was planted because it started all in a hotel with a hotel room number, and that is true. That was 9/11. So when you know about 9/11 in America, that's what people thought. This was all sort of like planted there. But this disease caused for many places in East Asia, particularly sort of like China, Hong Kong, Taiwan, Japan, and so all these areas, a huge angst. And they decided, we will want to be prepared for this in future. And so, and if people ask nowadays, why is East Asia and areas of Southeast Asia, why are they so much better off than we? Then part of that is because they had such a severe effect from this outbreak in 2003, and they planned with preparedness for another outbreak in the future. Another of these events was something called MERS. MERS stands for Middle Eastern Respiratory Syndrome, and this disease even has a higher mortality rate of between 35 and 65%. The current average is around 35% mortality rate, which is really quite high. And it came to, in South Korea, it spread from one case through a hospital, through a number of hospitals, and actually also infected a huge number of healthcare workers and led to the death of a number of people in that scenario. And again, South Korea said this will never have to happen, and we are prepared for this in future. And in 2014, actually it was December 2013, there was something happened in the border between Guinea and Liberia, where a lot of people lived, and there was a boy who was playing at that tree side, and there was some bats around, and ultimately he was the index case that then led to the spread of Ebola from West Africa. It was the Ebola Zaire strain, it was called. And in the past, all these infections caused probably the death of about few tens or few hundreds. This one caused the death of between 10 and 15,000 people, and it was the largest outbreak to date, and it led to enormous changes both in those areas and particularly in the Western world. So we built our current unit around that time in very short notice. It was built as a temporary unit. We are due for an upgrade and are hoping to have that in place in the next year or so. But we also, based on that, it was nationally identified that there should be an airborne network, and this is called airborne serious infections, or HCID network, as we call it. And we were one of 4 units that became one of those centres. There is Newcastle, Liverpool, and 2 units in the London area. Sheffield since March has joined this as well, so we're 5 airborne units now in the country. We had our first exposure to that then with a case of what's called monkeypox, which is a cousin of the smallpox virus, and it is not normally around in the UK. And we actually had the first UK-born case. That was a healthcare worker who infected herself from a patient that had come back from Nigeria. And of course, there were all these things then that the hospital gave me monkeypox. And again, that has led to that actually, personal protection or infection control, and the whole infection management is vital to how we deal with this. So coming to COVID, I suppose you may vaguely remember what was there in 2019. We could go to football games, have parties, celebrate, go on holiday, do whatever we liked. And well, has that changed? So it all started, actually, it was reported first on the 31st of December 2019, but we believe that the first case was actually sometimes in November arising, and then as soon as you have a few more cases, then it became known, and they reported this case in Wuhan in China, and this is why it was called, as derogatory by President Trump, called the China flu, or and this China virus. And actually, China has probably managed this subsequently in a very, very strict manner. And they have come out of this almost, although not completely yet. But it is shown that Li Wenliang, he actually was one of the whistleblowers from that hospital in Wuhan. And unfortunately, he died on the 7th of February of this year. And I remember how he put his pictures on Twitter. And that picture on the right-hand side that you see, that was his, I think his final picture that he put up. The world came together again, and particularly the scientific world, and they quickly identified ultimately that this was a coronavirus, and ultimately it was had some similarity to the SARS coronavirus, and that's why people got really concerned. But then they identified it was actually it was a slightly different virus, and that is important because ultimately SARS and SARS-CoV-2, as it's known, the COVID-19 virus has behaved differently to SARS. So SARS itself kills more people, but is not as easily transmissible as the SARS-CoV-2, which infects so many more people, and that's why it's becoming such a huge pandemic. You can see here on this picture, you hear in the news always about the R number, and there is a reproduction number, and we know that uncontrolled, the R number for SARS-CoV would be in the region of 2 to 5. So that is very high. Only measles, which is the most infectious virus that we know, has really a much higher R rate. COVID-19 presents really quite unspecifically, for most people, a bit like a flu-like illness, like a cold. And many, I presume, quite a few of you may have had COVID yourself, and some of you will have had absolutely nothing. And some of you may have had just a bit of like a taste of loss of taste or smell, but some of you may have had more symptoms. And so there's a whole spectrum there. Of the ones that become really quite ill, a substantial number then develop fevers, chills, cough, and ultimately become breathless. And on the right-hand side, you see cuts through what's called a CT of the chest, and you see some classical pictures, so quite a lot of damage at that time to the chest. And that's why patients become so breathless and so unwell. We know that if you're male, if you're older, if you're black or BAME, if you're from poorer and background where there is clearly not the same protection measures, if you have some medical conditions, particularly high obesity, but others as well, then your mortality goes up. I have added there also that if political measures are done by pure sort of like politics and beliefs rather than science, then we also have some problems, and I could add a few more people to that. So locally in January, we received the first 2 patients. At that time, they were treated as highly infectious. That meant we had a special unit. We had to dress in a very specific manner. It took a long time. One patient required 10 to 20 people alone to look after them. This all changed when it became clear that this was becoming a pandemic and the numbers would go up sky high. And we then developed an approach whereby simple protective measures, and sometimes when patients are on breathing machines, or then we need to have slightly more protective measures because the virus can be transmitted by both droplet and aerosol based. And doing this reduces the infection rates dramatically in healthcare staff. During the first wave, there were, however, big problems, clearly. The big 4 are that PPE was not always available, and you may have seen, some of you, those pictures where people use bin liners. Testing facilities were not available in the beginning. Care homes were neglected, and lockdown measures were either sometimes taken too late or wrongly, or there were lots of politics behind it. But all of this was to be done, and some of you may know that in the first wave, the hospitals were overwhelmed at the time very quickly, and so by lockdown, we achieved to reduce that. We now are in the second wave, and the Northeast and Northwest in particular have suffered a great deal. But I think we are with the lockdown measures in place, we are now again on a curve where the numbers are going down. Just what can we do? And this is sort of a reminder of when people say you should socially distance, so it's about space, it's about having mask wearing, and it's about, the overall contact, and it's washing your hands. All of these features are part of it. None of the features alone will be 100% to reduce the infection. And there's this Swiss cheese, the Emmental cheese type picture here. So if you take all those measures together, and some of them are personal, so that you and everybody else has to do, and some are shared responsibilities. So having adequate testing, having good ventilation in and outdoors, having the support, having quarantine measures in place, and clearly vaccine, that will help as well. So some final bits. I think we still have, this is still a new disease overall, and we are still learning. I have never seen in my career as a doctor so much research and so much publications going on in such a short time. all the statistics and all the epidemiology will still change. Good evidence is absolutely vital to get us out of this. Politics should follow the evidence rather than evidence follow the politics. Treatment and vaccines help, but are only one measure. So, and that's what we've really got to do. We're all looking forward to be on our own little island and not have any cruise ships around. But I think actually next year people want to go on holiday again. So I finish here with saying I can finally see some light at the end of the tunnel. Thank you very much. Hang on. So I'll hand over to, well, I think we'll probably just take the questions at the end. Is that correct, Laura?
Yeah, perfect.
I'll hand over to Ashley, who's a very good colleague of mine.
Hi, Matt, and you mine. Can you see my screen okay? Can you see my slides? Great. So I'm going to talk a little bit about the development of treatments for this new coronavirus. You know, we didn't know, is that okay, Matt? Can you hear okay? Yeah. So we didn't know anything about coronavirus, as Matt has said. So, you know, in terms of what it really did and how it affected us and what treatments would be useful. So in the past, you know, for example, HIV, we've developed really effective treatments for HIV that don't cure you, but do reduce the amount of virus in your blood and then lead to an immune reconstitution so that your immune system comes back and you are able to survive with HIV now for your whole life. Different viruses are different. So HIV is a DNA virus, sorry, an RNA virus is a retrovirus, which means it's RNA normally, but when it gets into the cell, it changes through reverse transcriptase to become DNA, and then that DNA is integrated into the your own DNA, and it lives there for the rest of your life like that, really within your cells, and then can replicate from the DNA that's been integrated into your own cell. Coronavirus is different. It's an RNA virus. DNA in human cells makes RNA that then makes proteins, and those proteins do things. In viruses are inert in themselves. You can almost say they're not alive, but when they invade into cells, they use your own cell to replicate, so that they're basically using your cell as a factory to make more viruses. When you've got an RNA virus, they do all this in the cytoplasm of the cell outside the nucleus, and basically they just make copies of themselves, RNA copies of themselves, and the RNA then is a template basically to make more proteins that will then make more viruses and will also help in that replication. So essentially, in terms of treatment, you know, classically with viruses, we've had treatments that have affected different parts of the either cell attachment of the virus to the cell or processes within the cell that help the viruses to replicate. And they're mainly viral proteins that we have targeted in the past. The problem with coronavirus is because it's a new virus, we have no idea, well, we don't know how to block those proteins. So it's you know, we have to learn how to do that for this specific virus. And we still haven't got a very effective specific antiviral against coronavirus. They are still being developed. So, you know, so this is a drawing that I did of the coronavirus back in January of this year, just as it was happening. I was quite kind of pleased to get this out on Twitter at that time, and as a drawing on the 31st of January. But actually, you know, my quote underneath this is this virus is going to keep us busy for a few more months. And actually, you know, that's going to be years. You know, you kind of think, well, these things might be over a little bit quicker. Maybe we'll develop treatments quickly and all this kind of thing. It won't be as severe as we thought. Actually, it is as bad as we thought. It's worse than we thought. It is still killing people in our hospitals now. And you know, treatment is really important. And I must say, I think all of us on here on this talk would say one of the most challenging things, and indeed one of the most exciting things about being an infectious disease physician at this time, is helping to develop those treatments. So I'm just going to go through this is a very complex slide, but essentially the coronavirus binds to ACE2 receptor, which is one of the receptors in our body that's used for different functions in our body. And I won't go into that in any detail here, but that binding is very important to the virus. And if we could find something that would block that binding, that might help. You then get replication within the cell. And there's various proteins. There's an RNA polymerase inhibitor, so RNase-dependent polymerase that could be inhibited by various drugs, and some of those are being explored. There's these proteases that help break down this, what we call a polyprotein. So when you first make a protein from the RNA, it's a very, very long protein. It's of no use to the virus. And the protease breaks down that protein into small blocks of protein that help the virus to replicate, and they're really necessary. And there may be protease inhibitors that may stop that process from happening, and that might be very good. But I think importantly, and what we're finding is that the host immune response is really key. There are clearly now, we know, two phases to the coronavirus. There's the initial viral phase where you get a lot of virus happening. There's a lot of replication, and there's possibly a little bit of cell damage happening there as well. And then you get a second phase that happens at about day 10. And those CT scans that Matt was showing were of CT scans in those later phases. And essentially, you may clear the virus on your own with your own immune system before about day 10. Most people who do that will stop having fevers, will be getting better. But those who are getting ill go on to get this severe sickness. And I think what we're finding, and as I'll go through, is that the immune system is really key in that. So I think one of the things to say at the beginning, when we first had the first patients, what do? We're doctors. We give treatment. We want to treat people. How do we treat people? And we were very, very keen to try everything and think, well, can we do this? The big problem with just trying everything is that might actually do harm to the patient, firstly. At best, it may be absolutely useless. And really, we need to do clinical trials to understand whether these treatments work. So hydroxychloroquine is an excellent example. That went into guidelines because we thought it might stop viral replication by causing what we call acidification of the lysozymes, where the virus is replicating and actually help to stop that replication from happening. And that was really thought that in the experimental experiments done in the lab, it was thought, well, this might work. But actually, when it's been taken out into the clinical environment in the studies that we've done, it doesn't work. And what you need to do is you have to do randomized controlled trials. And that's where you give a treatment to somebody, you ask someone for consent. This has to be done ethically. You have to consent someone into the study, or ask a relative if they can't, can you come into this study? And you either give them the drug or you don't. And you might say, well, goodness, we're not going to give treatment to people. Well, we don't know what works. So actually, and we know that these treatments potentially might cause harm as well. So we either give the treatment or we don't, and then we look at the outcome. So if the person has survived, that's one outcome you could look at. And you might say, well, if everyone survived in the treatment arm versus the placebo arm where we've not given, or the standard of care arm where we've not given any treatment, well, that survival will show you that treatment is working. Or you might say, well, people getting home earlier or coming off oxygen or not going to ITU, those are all what we call endpoints that you could look at. So that's what we did. And the first study that we looked at, one of them was antivirals, and this was with a drug called remdesivir. So this is a repurposed drug, so it's not specifically been designed against coronavirus, but we know it works to some extent against some other viruses, but it's certainly not perfect. So in Newcastle, we were approached to say, do you want to do this study? And we were one of 5 centres in the UK, one of around 30 to 50 centres in the world who did this study. And what we did is, was a double-blind randomized controlled trial. So when I consented a patient to the study, I didn't know whether they were getting the drug or not getting the drug. And I didn't know that until the results were released. So that made sure that I couldn't be biased and say, oh, this drug works. I think this patient got better earlier. And it's the kind of best type of trial really that you can do. So we did this trial here, and we were hoping it would work because it has some broad antiviral activity, and it has been shown to inhibit this RNA polymerase that we were talking about earlier, potentially. And what we showed in this, it was called the ACTT study, was that recovery indeed as an endpoint, it seemed to work. And we had to stop the trial early because they did this, they call it interim analysis, an early analysis to see if there was any harm or benefit, and it showed a benefit. So they said we have to stop the study because we're seeing a benefit here and this might need to go into treatment. And what we showed here, you've got the x-axis here is the number of days that the person's ill or in hospital, and then the proportion who've recovered against each day. And you can see here at around day 27, certainly you've got around 75% of people are recovered in the remdesivir arm, and lower, about 60–70% in the placebo arm, the arm where people weren't getting the drug. And that translated to an improvement from 15 days to 11 days in terms of recovery. So that's very hopeful that this drug may help. And you could look at this with if they were receiving oxygen, not receiving oxygen or receiving oxygen. Those receiving oxygen the benefits seemed to be better. And if they were on ventilated, if the patient had to have help breathing a tube down into their lungs, they were very, very sick. It didn't seem to help. But unfortunately, and possibly because we had stopped the trial earlier, but probably not, remdesivir didn't seem to help with survival. So even though this was an antiviral and it seemed to be having some effect, it doesn't help survival. So we are now giving this drug, but there has just been a study called a Solidarity study, which is a much larger study, but not double-blinded. So it was just what we call a placebo-controlled study, where they, or in fact, actually, I think it was even just standard of care versus this drug. And that has shown no benefit in survival either, but also no benefit in terms of length of stay in that study. So we're really not sure that remdesivir will work very well. And as I said at the beginning, that the virus is replicating very actively at the beginning. By the time you get the illness, it's probably not the virus that's the main driver of your sickness. So what we need to do is probably develop antivirals that we give very early in the illness, and that might improve things. And indeed, we're looking at whether or not giving remdesivir before someone comes into hospital helps to improve survival. And I won't go into hydroxychloroquine any further that we've talked about, but I think just to introduce the recovery trials. So one of the things that the UK has done really, really well was not to give people random treatments. So if you went into hospital in maybe Spain, Italy, France, you would be given hydroxychloroquine, but we had no evidence that it worked. So the recovery study was set up in the UK to look at whether these treatments worked. And basically they used different drugs. So you could either be randomized to getting hydroxychloroquine or an antibiotic that we think might help with reduce inflammation, or steroids. And other treatments have come along to put into those as well, versus just doing what we're doing, which is giving oxygen, supporting the patient, treating any complications that we know of. And what was shown here was that hydroxychloroquine had no effect on deaths. And you can see here that the percentage who died was even slightly higher than in those who had standard of care, although that wasn't what we call statistically significant. So it didn't actually kill you more, but it certainly had no effect on you. So then we come to this point, do other things help that will alter the immune system? So the most common immune modulator that we use in medicine, probably over all others, is steroids. We've had them since the 1950s, and they've been really important in making us able to treat some autoimmune diseases like rheumatoid arthritis, also really important in enabling us to do transplantation and so on. They were the first drugs used in those kind of situations. So the recovery study had steroids in as one of their arms. And you can see here, this is a massive study. 11,000 people went into this study. And in all hospitals across the UK, it was a very, very well done national drive to put as many people as we could into the recovery study, and has been hugely important in changing the care of patients across the world. And so we either gave dexamethasone or not. They were given these other drugs, as we said, in different arms. And we showed that basically around 2,104 within this trial received dexamethasone versus 4,000 approximately who had this standard of care, so who had nothing extra given. And dexamethasone was given at a low dose, 6 milligrams for 10 days. And as ID physicians, we all sat down and said, this is a bit crazy. We need to be careful about giving steroids here. It could make the virus go out of control. It could kill people. And this is why you have to do studies. And I must say, we were, if you like, proven wrong. But this is the whole point of doing research, it was shown the mortality was significantly lower in those taking the dexamethasone than usual care. And if you were ventilated, you saw a death rate in the usual care of 41% versus around a third, 30% in those taking steroids. And you can see these lines diverge, and the lower line is the dexamethasone with improvement. The only thing to be aware of here is if you gave the steroids too early, that might have then led to an increased risk of deterioration. So if the patient wasn't receiving any oxygen, then that wasn't a good time to do it, to give steroids. And that might be because you're suppressing the immune system just at the wrong time, so the virus can replicate more and then lead to more damage and more problems. The immune system is hugely complex, and Chris is coming on next, and he's going to explain this slide in full detail. I would not be able to do that. The immune system is very, very complicated, but it's just to say that there are a number of drugs that we are now looking at to see whether or not we can improve beyond steroids that immune modulatory effect. So the one that's looking potentially promising is this drug called toxilumab, and we may hear more about that. The other thing, the virus binds this ACE2 receptor, and that may have a part to play, or other things may, in causing clots. And we know that clots are a very important part of damage in this disease process. It's not just the virus, it's the way it affects your body. And this is always the case with viruses. And so we're looking at whether or not some of these things that reduce clots may work. And then we're looking at plasma on people. So what we do is people who've recovered from coronavirus, we get their blood, we then take the plasma out of that, and that contains antibodies, which we're trying to give to patients who are ill to see whether or not that will improve their outcome. And then we have some very clever companies have come up with what we call monoclonal antibodies. They've actually designed antibodies that specifically against that virus protein that binds to the ACE2 receptor to block that, to stop the virus from binding to the cell and getting in. And that's really, you know, I hope that's not being too complicated, but I guess the excitement and the, you know, one of the things about this is that we have in the UK really changed how we treat coronavirus. We've learned so much in the last few months to help us to improve patient outcomes. And I think we'd all say in the first wave, we saw a lot of people dying, and we saw some very young people dying. I think we are, and we need to see the data, but because of the treatments that we're using at the moment, we are seeing less people dying. And that required science to get there. And that we're going on to the vaccines next. So I think Chris is going to take us through those, I believe. Thank you.
Thanks, Ashley. Let me just find my screen. Okay, can you see this?
Yes, we can.
Yeah, great, cool. Okay, so we're I think we've got a great deal of time. So I'll go through this quite quickly so that we've got a little bit of time for questions at the end. So yeah, thank you, Ashley. Just going to talk about progress that we've made recently with vaccines against SARS-CoV-2. So this is a bit of a complicated picture, but essentially the message here is that Normally when we develop a vaccine, the process is to start after all the preclinical work is done to actually develop the product in the first place, to start with a phase 1 trial where we look at the safety of the vaccine in a very small number of people, so 10 to 50, and then a phase 2 trial, which is a larger group, maybe up to kind of 100 or so, or maybe a few hundred, looking at the immune response that's generated by the vaccine, and also again looking at safety. And that process maybe takes another couple of years, and then if it looks promising, it goes to a phase 3 trial, which is to continue measuring safety, but more importantly, to determine whether or not the vaccine is effective. And that process, again, can take another few years. Then at the end of that whole process, the data are looked at, they're submitted to the regulators, they make a decision about going ahead, and then the vaccine starts to be manufactured. That whole process conventionally takes about 15 years or so. In the COVID era, things are happening much faster. So as you've heard, we basically first encountered this virus right at the end of 2019. So we're nearly at the anniversary of its identification, and there are already phase 3 efficacy results on at least 3 vaccine candidates. So that is astonishing and unprecedented. And the word unprecedented gets bandied around quite a lot in the context of COVID, but this really is unprecedented. One possible way to interpret this would be this has all been done in a hurry. However, anyone who's worked on vaccine development will tell you that actually the reason that this process takes so long is not necessarily deliberate. It is quite often because of problems with getting funding, the speed at which regulators look at trials, and how the kind of vaccines progress through. And what we know from the experience with COVID is that if enough political and kind of societal will is there and enough money is there, that these things can be done in really quick time. So it's actually a pretty impressive feat. This is the beast, the SARS-CoV-2 virus, as you've heard about. All of the vaccines are designed to target this thing here, which is called the spike protein. This is the protein which the virus uses to enter into and thus infect cells. And vaccines are designed to induce an immune response that mimics what we normally produce after we've experienced an infection, but without actually having to get the infection. So the purpose of a vaccine is to mimic the immune response to the infection. And in the case of a SARS vaccine, what we're aiming to do is induce two things, what are called virus neutralizing antibodies. So these are proteins that bind to the viral spike protein and block it from binding to the host cell receptor, and therefore stop the virus infecting cells. And also these things called T cells, which are immune cells that recognize cells which have been infected by a virus and then kill them. And that allows prevention of infection to take hold and spread. The T cells also are really important in helping to make these antibodies. And so T cells are an important part of an immune response to any infection. There are various platforms being used at the moment. I'm just going to focus on a few of these. So these are different ways to trick the immune system into thinking that the person has had the infection. So the old-fashioned way is just to take the virus, grow it up, and then inactivate it with whatever process you use. That can be ultraviolet light, it can be fixation with chemicals, whatever. And then you use the inactivated virus and you inject that into somebody in order to make an immune response. Then there's a slightly less sophisticated method, which is to make the recombinant protein and then inoculate people with that. And then there's increasingly more sophisticated methods, and these are the ones that have been in the press in the last month or so. So this is a very new method of vaccination where you use what's called mRNA that Ashley talked about earlier. So this is the message that tells a cell to make a protein. So in this case, it's a little bit of message telling the cell to make the spike protein, so that makes the cell look like it's been infected and the immune system responds appropriately. And it's just encased in this little lipid droplet, which allows the lipids to fuse with the cell membrane and get the message into the cell. The other way to do this is to use another virus, in this case, which is usually a common cold virus called an adenovirus, that infects cells very efficiently, and it doesn't grow itself, it doesn't replicate or cause disease itself. All it does is it makes the cell make the message for the spike protein, and so the cell makes the spike protein. So they're all different ways of trying to make the immune system recognize the viral spike protein. And this is just to kind of give you a bit of a summary of how many different vaccine platforms are currently in development. It's absolutely enormous. So there are hundreds and hundreds of different candidates at the preclinical phases, all the way through, and in fact, 3 vaccines which have released efficacy data in the last 3 weeks, I think. And it makes sense to focus on those. So I'll very, very briefly describe those 3 vaccines that you have seen in the news and what we know about them and their behaviour. So the first is this impossible to remember name, which is what all pharmaceutical companies use to describe their products, but it's made by a company called Pfizer, so it's known as the Pfizer vaccine. And this is the results of the phase 1 trial. So this is where the goal is to assess the safety and also the ability of the vaccine to make an immune response. So the symptoms that participants experienced after injection of 2 doses of this mRNA vaccine were pain at the injection site, which was dose dependent, and a little bit of other local symptoms, and then some systemic symptoms, which are very well recognized with all sorts of different vaccines, and effectively just mimic flu-like symptoms. So they normally last for about 24 hours or so and then settle down. So all very kind of standard. This is a kind of important bit in relation to whether or not this vaccine is going to be any good. And this is a complicated slide, but it just summarizes the immune response. So in the vaccine dose that they ended up selecting for the efficacy trial, the participants get 2 doses, and these are the immune responses. So this is the immune response in terms of antibodies produced after 1 dose and then boosted after 2 doses. And this, in comparison, is the amount of antibodies that patients make after they've had coronavirus disease. So you can see that the level of antibodies made in both participants under 55 and in older age groups is way, way higher than what we normally get after an infection. So that's obviously extremely encouraging. And what that translated into, as you probably know, is efficacy. So in the very, very large phase 3 trial, which is designed to look at whether or not the vaccine works, there were around 40,000 participants who received 2 doses of either vaccine or placebo, and a placebo is just something which doesn't have the same effect but is there as a control. And a decent proportion of those participants were over 55, which is a group that's at particularly high risk of complications of coronavirus infection. And there were very many more cases of COVID-19 in the recipients of the placebo than in the recipients of the vaccine, and that translates into 95% efficacy. And basically what that means, when we talk about vaccine efficacy, we talk about the ability of the vaccine to prevent the disease under kind of perfect conditions, so in the setting of a randomized clinical trial. And that's different from the term effectiveness, which is what we use to describe how good the vaccine is at preventing disease in the real world, where conditions are imperfect. In other words, things aren't done according to the very, very high standards in clinical trials. And the way that we calculate vaccine efficacy typically is by dividing the risk occurring in the population that have received the vaccine with the risk of disease in the control population. So that's those who've received the placebo. That's known as the relative risk. And the way it's expressed is just 1 minus the relative risk as a percentage. The other one that very recently was announced is the same technology. So I won't linger on this one to any great degree. Again, an mRNA vaccine given with 2 doses made by the American pharmaceutical company Moderna. Again, this is quite conventional reactogenicity, so symptoms of flu-like symptoms, and also pain at the injection site occurring in most participants that received the vaccine. Again, most of these resolve within a day or 2, so are quite standard. And again, you'll be used to looking at this now, but these are the antibody titers. And so what we can see with the different doses that they used, that the antibodies were an order of magnitude higher than what are normally made by patients. So again, suggesting that this will be effective, and as I kind of alluded to, again, similar to the Pfizer vaccine, the number of cases of COVID in the placebo recipients in this large phase 3 trial was much higher than in the vaccine recipients, again, translating into 94% efficacy. And then the final one I wanted to mention, which you probably heard about at the beginning of this week, is the chimpanzee adenovirus vector vaccine produced by University of Oxford in partnership with AstraZeneca. And we've actually been running this clinical trial in Newcastle, the phase 3 component of it, since around about May, June of last year. Again, a very similar safety profile with local injection site reactions in the recipients of the coronavirus vaccine. In this case, in this trial, instead of a placebo, so instead of salty water, the participants actually received as a control a different vaccine, so in this case a vaccine against meningococcus. And so the coronavirus vaccine was a bit more reactogenic than the meningococcal vaccine, but not by much. And again, 2 doses of the vaccine led to the production of good levels of antibodies, which were in the range of what patients who have recovered from coronavirus disease develop. And you'll have seen this in the news in the last couple of days. So they very recently released interim efficacy analysis of this vaccine in a slightly different fashion than in the other 2 studies. There were 2 different doses trialed in this study. That wasn't actually intended at the beginning of the trial, but there was a problem with how they measured the amount of virus present in the doses, so that a group of participants received a lower than planned dose for the initial dose of vaccine, but the regulators thought it would be sensible to continue with the study to compare these 2 different doses. And actually that was quite sensible, because the vaccine efficacy overall was about 70%, but it was actually higher in the participants, oddly, that received the lower dose priming and the higher dose boosting. And the reason for that is not known. Again, all of these reports are based on press releases, and none of these vaccines have yet been published, so the data aren't available for us to scrutinise. So what we know now, considering that this is only 11 months down the line, is that measures of immunogenicity that have been identified in early phase trials appear to translate into excellent levels of vaccine efficacy. There haven't been any major safety concerns with any of these vaccine platforms, and they're providing extremely high levels of protection against severe disease. And to be perfectly honest, this is way beyond what most of us expected to be the outcome of this, because if you compare the efficacy of these coronavirus vaccines to vaccines against other respiratory viruses, such as flu and RSV, it's extremely impressive levels of efficacy that are being achieved. So I think these results from these first 3 studies to report bode extremely well for the many other vaccines that are in development, which are all inducing pretty good immune responses. And it's likely that a lot more of these vaccines that are being developed will show similar levels of efficacy. So the other things that need to be considered when we're weighing the relative benefits of the different vaccines, as more and more of them become, reach their primary endpoints and start to be reported and then potentially licensed, is the cost. Things about the logistics. So how do you store these vaccines? How are they transported around the world? And how feasible are they to be given to populations who need them? And also, what's the capacity to actually make large numbers of vaccines, considering the fact that coronavirus is a global pandemic and there's hundreds of millions of people who have yet to be exposed to this virus. Just to bring it back a little bit closer to home, I thought I would mention what's actually happening in Newcastle in relation to vaccine development. So as I said, we've been conducting the Oxford AstraZeneca vaccine study and recruited 700 participants into that. We're starting a phase 3 trial of another vaccine candidate made by the company Janssen next week. And then we're starting another one at the beginning of January, looking for healthy volunteers for a phase 1 trial. So that's the earliest phase to test whether or not the vaccine is safe and induces an immune response. And if anyone's interested, as a little plug in participating in any of these studies, then have a look at these links or look at Research Plus Me on Twitter. So I hope what you've taken from that is that vaccine development is proceeding at an unprecedented speed. Newcastle is playing a central role in that process, and the results so far are extremely encouraging, but present us with actually enormous logistical challenges that I'm not sure that we necessarily anticipated having this quickly. So we now have to work out how we're going to give hundreds of millions of people vaccines as and when they become available, and how to do that in a safe manner. Some outstanding questions still to be dealt with. We don't know much about whether the efficacy is against clinical disease or whether it's against infection, and some of the studies have looked at that and some of them haven't. So that's important because there's a lot of asymptomatic infection that happens, and it would be helpful for a vaccine to protect against that, because that will prevent transmission in the community. We don't know much about how good these vaccines are in people who are particularly high risk, because often they are excluded from these types of clinical trials. So those studies in high risk groups need to be done. And we don't know much about how long the protective effects of these vaccines last for. So that's another important question that we have to follow up on. So I'll leave it there. This just acknowledges the various people that have been involved in doing the vaccine work locally. And I'll also thank Matt and Ashley for their earlier presentations, and I think we've got a little bit of time for questions.
Excellent, thank you very much. Thank you to all 3 speakers. That was a really interesting topic and pitched at the right level, because I understood, which is saying a lot. So I know that we are technically at the end of the session, but we've still got a lot of people online. So what I've done is I've picked a selection of questions, and we'll just have 5 minutes or so for questions at the end. So if you're okay, I'll just fire away with the first one, which is, would you have the vaccine, and how trustworthy is it?
Yeah, I think that is for Chris.
I can take that one first. So the answer to the first bit is definitely. And what was the second question?
And how trustworthy is it, or will it be?
How trustworthy is it? Yeah, I mean, I think the way that vaccine trials are done is extremely robust and with a huge amount of care and attention to the safety of the participants. And these trials are overseen by very, very serious regulators who are extremely careful to make sure that everything is done absolutely perfectly. So that's why there is a difference between what we would describe as efficacy and what we describe as effectiveness, because clinical trials are performed to extremely high standards, as we all know, having done them. So the results are absolutely trustworthy. I think what sometimes happens is the way that things are reported in the media maybe suggests that there are issues, and I know that there's been some discussion around the kind of way that the different doses in the Oxford AstraZeneca study were presented, but actually that was all done quite openly with participants all the way through. It's just that conventionally one doesn't report the ongoing process of a clinical trial in the media, so that's why these issues weren't necessarily put front and centre of a press release.
Perfect, thank you. What lessons do you think have been learned from this pandemic?
Well, that's a good one. I think we're still learning lessons. From my point of view, is the biggest lesson is that we have to be prepared. We have to be better prepared in future. I think we went off the boil after 2015, and clearly investment wasn't made. And I think the other one is what lessons we've got to respect each other more and be tolerant of each other more. And it's not about me. I think wearing a mask is about, for instance, at the moment where people are saying, oh, I don't want to wear a mask, I can't wear it, and so on. It is about you're protecting somebody else, potentially, or reducing the risk to somebody else. So being tolerant and showing respect to somebody else is an important part. And if we can do that, and we're becoming a little bit more human again, we have lost that over the last few years. That's what I think.
Perfect, thank you. You got something to add?
Yeah, I would probably say the other lesson that we've learned is actually that that the scientific community can actually do a lot to help society when it works together and confront something like this. I think it's a kind of bit of a political cliche sometimes, but there's no doubt that the progress that science has made over the last year has just been astonishing, and it's very unusual to be in a situation where you've kind of got knowledge of the biology of a new virus. You've got effective treatments for patients in hospital, and you've got multiple effective vaccines all available within a year. So I think one of the lessons will be we need to be prepared for new viruses and new pathogens, but we can use the lessons we've learned from this about working together and working collaboratively to combat them.
Yeah, I'd echo both Matt and Chris's comments there. I think that's absolutely true. I think the one worrying thing, though, is that we had SARS-CoV-1 and we learned nothing from it. If we'd have really, gone and understood that disease, we may have been able to help to make this a less dangerous thing. But, we like to look at the best of things, don't we?
I think I want to point out to that. It is Taiwan, for instance. They really feared SARS in 2003, and they decided to, invest into PPE and into facilities and into real preparedness. They could, within a month, quintuple their output of PPE, which was unheard of. Likewise, South Korea, they had, after MERS, they developed a system of where they could fast track technological advancements and developments. And so to overcome the hurdles of normally, not just vaccine takes years and years, but technology and diagnostics and takes forever. And they did it in, it started, and within a month or two, they had new technologies developed. And that is certainly something that the Western world has to learn. And finally, I think we've got to learn sometimes from our own mistakes. We are all this about freedom, and we want to, and sometimes, as I said, we've got to respect how we deal with that freedom to not actually harm others.
Thank you. A question here, how does being from a BAME background make an individual more susceptible to COVID-19?
So we don't know many aspects still. That is absolutely true. There are likely some genetic factors that are part of this. And that is the most important answer. What may be part of it is that there are some often people from a BAME background or many BAME live in more cramped environment. So there may be more people. There may be socially interacting more with all the family members from all ages, and that may have been a social, cultural type. We know that some of the BAME population are from poorer background as well, and that may have had an effect on it as well. So that their living conditions are not as, are not from middle class, bigger environments of nicer buildings, and so that may play part of it. But the majority, we truly don't know.
Thank you. And I think we'll make this the last question. I hope you're not going to ruin everyone's Christmas here, but do you agree or disagree with the COVID restrictions being lifted for Christmas?
Yeah, so I think I'd like to just sort of put this in, because there's been a few as well, questions about the seriousness of this disease as well, which I think that ties in with this idea of should we be closing Christmas down? And I think they're absolutely right. People need to ask these questions, because you guys are the least likely to get ill from this, and you may well have not seen anyone who's ill. But what we've seen as healthcare professionals is nothing like we've ever seen before. There's people who say that this is a bit like a bad flu. Well, when we had the swine flu pandemic, it was nothing like this. And what we're seeing is a lot of really sick people. The youngest person I've had in the last 2 weeks was a 33-year-old who I think in the first wave may have had to have at least gone to ITU. I think the youngest person we've had who's died in Newcastle is a 26-year-old. I may be wrong, but I think that's right. And then, it's devastating to anyone over the age of 70, certainly over 80. And if you're a nursing home resident, some nursing homes had 50% of their nursing home residents die from this. So, I'm not putting down what you're saying. In your age groups, it isn't that serious a disease. There's still some people who get very sick from it, though. And the risk of you getting sick is very, very low. But I think what I'd say is we do need to protect our older individuals. So my concern is, I have children and they go to school, and there is a potential. We don't think any of us have had coronavirus. I know I haven't from my last tests and antibodies I've had. And if I got it and then passed it on to my mother, who's over 70, the risk of dying is, relatively high. And so that's the concern I have about mixing over a Christmas. I think we then may see a third, if you want to call it a third wave, but it'll be a whatever wave after Christmas. And I think that is a likelihood. And it's a very sad thing, this disease. It's certainly causing disease that's being transmitted from different communities and from different people. from the young to the old, even the old to the young, and but it's affecting certain individuals in a much more severe way. And it is a sad thing. We've just had in the last week, one lady died, and her son died in the same day. And these are really sad things. I've never been more saddened as a health professional by what I'm seeing on the wards. It's been very emotional for us all.
Yeah,
I can only. Sorry, you go first, Chris.
Yeah, I would just a kind of short one. So I think that the risk with opening up for Christmas is that we compromise the control that we've got. The downside is obviously that we lose the kind of cooperation of the population at a time when we don't want to do that. So it's a very difficult question. There isn't a right or a wrong answer to it. Personally, we're just putting it off. So I've told my parents to stay away, and I'm not going to get anywhere near my grandmother until they've had a vaccine. Once they've had a vaccine, then and I've had a vaccine, then it will be fine. And it makes no sense to me to throw away all the progress that we've made at the point where we're just about there, but we're not quite there, irrespective. And we've, nobody was making a song and dance about not having Diwali. So why should Christmas be any different?
Yes, I think every Diwali is an important celebration. I think Christmas is just a step up in overall in the UK, and therefore it holds more emotional sort of like baggage overall. There is a real risk. I think that just personally, whilst I haven't often agreed with the government over the last 10 months in their decision processes, but for instance, you've heard today that Newcastle or a lot of areas have remained in what's called Tier 3, which is almost like a type of continuation of the lockdown, just a prolongation. We have seen in Newcastle, it's not seen everywhere in the country, I have to say, or even in the Northeast, but in Newcastle, we've seen that figures are coming down. So last week we had around 1,200 cases in Newcastle. Or in this last 7 days, we've had 800 cases in Newcastle, and the week before we had 1,200 cases. So there has been a reduction overall in numbers, and our admission figures in hospital, or our figures in hospital, have come down by about roughly 25 to 30% from the height about 10 days ago. So there are some positive signs. So a prolongation will help further. I do think that people can create sort of like safe bubbles. So over the Christmas period, but they can't just go say like, oh, normally we go and party all the time and we get all the families together and have just be that. You really have to be aware of, all of us who've worked with patients and have seen patients suffer, and it is not just the deaths and in patients. It's also about a lot of people, or some people have suffered and suffering for prolonged periods with fatigue, tiredness, with weird, strange symptoms that we can't even yet fully explain. And I think if we can all, again, show a bit of respect and just, be together, but in a more safer way, then you can have some Christmas celebration. If we had gone into a much lower tier and gone out, I would think we'd be actually filling up rapidly at Christmas already again. So we have an opportunity. The government was asked today, oh, are you doing this so that you can you're preparing then that the Christmas figures afterwards are going up? There may be some more cases. And as Chris said, we need the vaccine, but the vaccine will take time. None of the whole nation will not be vaccinated over the next 3 to 4 months. It will take longer, I think, and we still have to include our social distancing and our measures about face mask and hand washing and just a bit of respecting people.